Hi everybody, Miss Calabrese here. Welcome back to your microbiology lecture series. So in this video, we're going to be talking about cell transport. So that means how things get in and out of cells. All right, so we learned in the last video that the cell membrane or plasma membrane is a selectively permeable barrier. That means that some things can pass in and out easily, some things are going to need to be transported actively, and some things are just not permitted to be allowed in or out of the cell. All right, so there are three major categories here for cell transport. Those are passive transport, which requires no energy, active transport, where we're moving things against their concentration gradient, and that does require energy. And then bulk transport, this one is movement of things that are just too large to move otherwise. So this one is not necessarily related to concentration gradients, but just has to do with the size of the thing itself. All right, so let's start off by talking about different types of passive transport. So passive transport it is embodied by the concept of diffusion. So diffusion is just the idea that particles want to move from high concentration to low concentration. So that means they want to move down their concentration gradient. So you can think about it as balls rolling down a hill. They just want to move down to that lower concentration gradient and be more spread out from one another. So the way to demonstrate this is, for example, if you spray perfume in one area of a room, initially those little perfume molecules are going to be all kind of closely aggregated and directly in the area that you sprayed them. But if you give it enough time, those perfume molecules will separate themselves from one another. They will diffuse out and eventually spread out through the whole room. They won't be as concentrated because they're going from an area of high concentration altogether to an area of low concentration being very spread out. So this kind of diffusion doesn't require any energy. This is how molecules normally want to move. And so we can have this kind of diffusion happening across cell membranes. So sometimes if we have a high concentration of a particle on one side of a membrane and a low concentration on the other, the particles will naturally flow down their concentration gradient until it's even on both sides of the cell. Right, so that's just normal diffusion. Facilitated diffusion is basically that same concept, except instead of having to diffuse kind of in between the phospholipids of the membrane, for facilitated diffusion, we essentially open a door to allow that diffusion or facilitate that diffusion to happen more quickly. So an example of facilitated diffusion are structures called aquaporins. So aquaporins are basically doorways that allow for the fast diffusion of water across a membrane. All right, so here's what diffusion looks like. So in this picture A over here, we can see we've got a membrane here, so a selectively permeable barrier. On one side of the membrane, we've got some red dye, and on the other side of the membrane, there's no red dye. Right, so we've got a high concentration on one side and a low concentration on the other. Given enough time, if that dye is able to cross that semi-permeable barrier, eventually we'll go from having a high concentration on one side and a low on the other to being even all the way throughout. So we've lowered the overall concentration by allowing those particles to spread out and get maximum space in between them. Right, so this is, again, this is normal diffusion. This doesn't take any energy. It's a natural process. All right. Another type of passive transport we call osmosis. So an osmosis is basically a specific type of diffusion. So it's a subcategory of diffusion. Specifically, the definition is osmosis is the diffusion of water across a semi-permeable membrane. So specifically the diffusion of water specifically across a semi-permeable membrane. That's what osmosis is. And so a lot of times osmosis we see happening when that semi-permeable membrane is in place and it will allow water to pass through, but not allow other particles to pass through. So for example, in this picture here, we've got a flask that's sitting in a beaker of water here. inside that flask, we've got a solution that's got 10% solute, so 10%, let's call it green dye molecules on the inside of that flask. So we can see that those green dye molecules are in high concentration on the inside of the flask and low concentration on the outside of the flask. So what's going to happen here is you would think, okay, those green dye molecules want to go from high to low concentration, so they'll just cross the barrier and go into the beaker. But if that semi-permeable membrane prohibits the movement of those green molecules, that means the only thing that can move is the water. So the water will actually cross the membrane in the opposite direction. The water will flow towards those green molecules to try to dilute them. So we'll see water kind of going upwards into this flask. So eventually, see the water line here is about halfway up the cylinder, and the water line here is almost at the top. So what's happened is because the green molecules cannot cross the selectively permeable barrier and the water molecules can, the water molecules will rush inside that flask to try to reduce the concentration of green molecules inside there. Right, so that's the concept of osmosis. So the water is moving across the semi-permeable membrane to even out the concentration gradient, even though the other molecules are unable to move. All right, so this concept of osmosis is going to be really important to understanding how cells work, whether those are bacterial cells or cells inside your own body. Right, so this is particularly applicable to your own red blood cells. So your red blood cells are floating around in your bloodstream. They want to always be in a situation where they've got the same concentration of particles on the inside and on the outside of their cell membrane. So when we have the same concentration on the inside and the outside, we refer to that as an isotonic solution. So isotonic solution means the concentration is the same. So same amount of solute on either side of the membrane. And for the cells in your bloodstream, that's about 0.9%. So 0.9% salt on the inside of the cell, 0.9% salt on the outside of the cell. And that means osmosis is going to be happening, but it's happening in both directions equally. So the same amount of water is entering the cell as is leaving the cell. And so the cell stays perfectly isotonic, perfectly happy at all times. All right, if we change that concentration a little bit, so if we have a higher concentration of water on the outside and a lower concentration of solutes on the outside, so that means we have more solutes on the inside of the cell, we refer to that as a hypotonic solution. So hypotonic means more water and less solutes on the outside than on the inside of the cell. So what's going to happen here is essentially if you've got a higher amount of salt on the inside of the cell, water is going to try to rush into that cell. So it's going to head in the cell to try to dilute the salt that's on the inside of the cell to get us back to an isotonic concentration. The problem is there's only so much room on the inside of your cells to take on water. So if it's too salty on the inside, not salty enough on the outside, and water is rushing in to even that out, we could possibly get in a situation where we start to see lysis. So to lyse means to break apart. So if your cells take on too much water because they're in a hypotonic solution, we risk the possibility of lysis. Right, the opposite situation there would be a hypertonic solution. So hyper means more. So, and this is referring to more solutes on the outside of the cell. So this would be, for example, if you drank a lot of salt water, right? There's a reason you don't drink water from the ocean. It's because it's way too salty for you. And it would put your cells in a hypertonic situation that looks like this. And in this case, since there's more salt in the solution outside the cells, water is going to be leaving the cells. So water will cross the membrane and leave the cell to try to dilute the solution on the outside, which is not great because we need that water on the inside of the cells, right? We need to maintain an isotonic concentration. So in this case, the cells will shrink if we put them in a hypertonic solution. So hypotonic, they expand. Hypertonic, they shrink. All right. So those were all conditions of osmosis. So now we're going to move on to active transport. So active transport is transport that takes a little bit of energy. So all of those diffusion, osmosis, facilitated diffusion, that was all passive, no energy involved. Those are natural ways that particles just want to move. Active transport is what we need to do when we have to go against the concentration gradient. So if we're going against the concentration gradient, that means instead of going particles moving from high concentration to low concentration, which I like to think of as kind of rolling down a hill, right? It's really easy. Against a concentration gradient means like you're going from low to high. So you're starting off with a low concentration, try to write here, low concentration up to a high concentration, and that's like climbing up a hill. So climbing up a hill takes more energy than falling down a hill, right? So that's why we refer to this as active transport. Active meaning we have to supply some energy, and that energy is going to be in the form of ATP. So ATP is your cell's molecule of energy. So we would do this, for example, in this picture, you're seeing looks like some potassium ions. So we've got a low amount of potassium ions on the outside of the cell and a high amount of potassium ions on the inside of the cell. If we continue to move potassium ions in, we're creating a higher concentration gradient. So we're going against the gradient and creating an even higher gradient, and that takes energy, right, to move things against their concentration gradient. So this is a really important concept that a lot of your cells use a particular type of active transport called a sodium-potassium pump. That sodium-potassium pump is going to be important in maintaining a concentration gradient across the membrane of the cells in your body. Okay, and that brings us to our last type of transport. So last type is bulk transport, and bulk transport doesn't have anything to do with concentration gradients. It basically just has to do with moving things across the cell membrane that are too big to fit through a pump, a protein pump. So these are, in this case, we use vesicles. So I mentioned vesicles in the last video as being kind of like a little bubble of phospholipids that is able to carry things around on the inside of your cell. But those vesicles can also be used to bring things in from the outside and also push things out from the inside. So let's look at an example of exocytosis first. So exocytosis is moving large things out of the cell. And so here's what that looks like. So I've got a vesicle here, so we can see this bubble of phospholipids carrying some particles inside here. That phospholipid vesicle is then going to merge with the cell membrane and eventually open up and release those particles into the external environment. So that's what exocytosis looks like. So exocytosis is letting those particles leave the cell through vesicle formation. Right? And this takes energy because look what we're doing here. We're changing the shape of the membrane, right? So the membrane has changed shape here. And remember, membrane shape changes come from microfilaments associated with the membrane, and that takes energy, right? So that's why this is an energy consuming process. Right, so endocytosis is the opposite. So endo means within. So this is bringing stuff into the cell through vesicle formation, through change in the shape of the membrane. And we can do this in a couple different ways. So there's some subcategories here. So phagocytosis is when we engulf large particles. So example of this would be, this is how your white blood cells eat bacteria, is by phagocytosis. So they wrap themselves around a bacteria, eat it, bring it into the cell. Penocytosis is same process, but usually for smaller particles or liquids. And then the last type here, receptor-mediated endocytosis. This is a specific type of endocytosis to bring in only special types of particles. All right, so here's what endocytosis looks like. So it looks like the exact opposite of exocytosis. So we've got some particles here that we want to bring into the cell. we're going to kind of wrap our cell membrane around that until we've created a vesicle, and then we can bring that vesicle to the inside of the cell. Right. And in the example of receptor-mediated endocytosis, so what we have here is referred to as a coated pit. So this is a surface of the membrane of the cell that's got specific receptors on it, and those receptors are only going to trigger vesicle formation for particular molecules. So one of the most common types of this is for cholesterol. So for your cell, in order for your cells to take up cholesterol, the cholesterol molecules have to hit the receptors in these coated pits, and then that'll trigger vesicle formation. And here you can see the vesicle form that's going to take that cholesterol inside the cell. Okay, so of all the different types of transport we just talked about, which ones are going to be considered passive? All right, and there's your answer. So diffusion, osmosis, and facilitated diffusion are all considered passive. Endocytosis, exocytosis, active transport with a protein pump, those would all be considered active. All right, so I hope this helped you understand cell transport. Let me know if you have any questions.